Righty ho, everyone. So welcome everyone to this morning's BREEDPLAN webinar. My name's Gudex. I'm a member of the Extension and Engagement team based in Armidale, New South Wales. And I'm joined today by Catriona Millen, who is our team leader based in the same location. And Catriona will be delivering today's webinar with me doing the background stuff. So as most of you will be aware, we've been doing fortnightly webinars and we've done 4 already. The videos will be made available or already have been made available. Catriona, sorry, I don't know.
Have been made available.
They have been made available. And so today's one is an introduction to Single-Step BREEDPLAN. And the next one on the 26th is BREEDPLAN into the future. And these are all being done at 7:30 p.m. GMT or 8:30 p.m. CET. So just some housekeeping. Please make sure your microphones are on mute, which I see they all are. It is being recorded and we will provide a link in the future. So if you've want to go back through it. And if you have any questions, please use the raise hand function at the bottom of your screen or type into the chat there any questions, and we will get to them and hopefully be able to answer them for you. So with that, I believe it's over to Catriona.
Thanks, Boyd. So this evening, sorry, it's still morning for us here, so we will alternate between those. But this evening, I'm going to talk about what is genomics, what is single-step BREEDPLAN, what are the benefits of single-step BREEDPLAN, take you through a couple of case studies that show the impact of single-step BREEDPLAN for some of the breeds that have been using single-step BREEDPLAN for a number of years now. And then finally end with some considerations. So we'll talk about whether or not performance recording is still important. And I just wanted to touch on which animals to think about genotyping. Okay, so we use the word genome quite a lot. And what it means is the genetic material of an organism. So it's all of an animal's DNA. And so you have your animal here. That animal is made up of cells. Inside the cells are the chromosomes. And the chromosomes are where the DNA is stored. So we're talking about at a cellular level, looking at the genetics, all of the DNA of the animal. And what we've seen happen more recently is the ability for beef producers to take a tissue sample of some description from an animal and send it to a commercial genotyping lab for a DNA test. Now, here in Australia, there's 3 sample types that have been used commonly. Historically, we used blood as well, but more recently, we've seen tissue samples taken with a TSU or a tissue sampling unit, hair, and semen. And there's some pros and cons of all of those. So if we think about the TSU or the tissue, that's taken as an earplug sample, often in conjunction with calf tagging. And the good thing about TSU samples is that they can be collected from birth, but the downside is that you do require a specialised applicator gun and need to purchase the TSU tube. So you can see the applicator gun down here and the tubes there. Hair is taking a hair sample involves pulling approximately 40 hairs from the tail switch of the animal. And if you are taking hair samples, it's really important that they're pulled, not cut, because the hair is actually contained, sorry, the DNA is actually contained in the hair root here. So you need the roots. The advantage of hair samples is that they are probably the best sample type that is suitable for long-term storage at home in a room temperature environment. The downside is that the genotyping labs have told us that they recommend that they're not collected on calves younger than 60 days of age. And that's because those hair roots aren't necessarily fully formed in a young animal. and they can't guarantee that they'll get DNA samples when they do the DNA extract. But if you are looking, and we do recommend considering having a backup sample of all of your animals, then one thing that is suitable to use there is to pull some tail hair. So even if you are taking tissue sample units, we would recommend that you pull some tail hair as a backup, store it in a paper envelope, not plastic, because it will sweat in plastic, and put it in the cupboard, clearly labelled with the animal's ident, and that way you've got a backup sample if you need one in the future. Our third type of sample that a lot of producers use to get DNA is semen straws. The advantage of this is that you can collect or use them to collect DNA samples when the physical animal is unavailable. So that's quite useful if you're using a sire, for example, an AI sire from overseas, or if you've got an animal that is physically deceased, you can still go back if you've got a semen straw and get a DNA sample. Obviously, technology only available for mature bulls. So what are the major applications of DNA technology in cattle breeding? Well, there's really 4 major applications. The first is parent verification. The second is to help manage genetic conditions. I wasn't going to touch on this too much today, except to say that we find genetic conditions in all breeds, not necessarily the same genetic condition, but it's not restricted to a single breed. There are genetic conditions across all of the different breeds of beef cattle. And for many of those genetic conditions, there is a DNA test available to help beef producers identify whether the animal is affected, a carrier, so unaffected but carrying a copy of the genetic condition, or unaffected. And that's important because most of these are inherited in a recessive manner where you need 2 copies, so one from mum and one from dad, in order to see the genetic condition passed on. And so what you want to ideally be doing is avoiding carrier to carrier matings. And if you've got a genetic test that tells you whether or not the animal is a carrier, that means you can make more informed selection decisions about whether or not you should mate 2 animals together. There's also use in managing qualitative traits. So this is things like coat colour. So black and red being the base coat colours is a good example. There's a DNA test for that. And also horn or poll status. And there are DNA tests for that as well. And then finally, DNA is also useful in genetic evaluation. And so in BREEDPLAN, we've termed when we use DNA information, single-step BREEDPLAN. So they're the 4 main applications, but in reality, most beef producers will be doing a DNA test for a combination of those reasons. And so certainly here at home in Australia, we've seen breed societies introduce bundles where you nominate with your breed society that you would like to do a DNA test and its purposes, and it'll include PV, use in genetic evaluation if you've got single-step BREEDPLAN, and also any genetic conditions that are found in the breed. So we're seeing, rather than producers having to nominate, 3 or 4 different things that they'd like done, many breed societies will offer the bundle where it's all included. Okay, so single-step BREEDPLAN. Currently, we've got 17 single-step BREEDPLAN analyses. The first of these was implemented back in 2017, and we've got 5 single-step BREEDPLAN analyses that include breeds across the UK and Europe. So Charolais in Hungarian Charolais, they first implemented single-step in August 2022, as did South Devon in the UK. They both went up in the same month. And since then, UK Hereford implemented on January 2023, UK Beef Shorthorn, December 2023, and Aberdeen Angus in the UK, February 2024. So that's roughly 2.5 years. In the last 2.5 years, we've had a number of UK and European-based breed societies implement single-step BREEDPLAN. But we're certainly working on having more in development. And we've got several breeds, 2 to 3, that we are hoping will implement shortly. So this list will be ever expanding, and we are certainly working with breeds to encourage the collection of a reference population, which I'll talk about in future slides, so that we can expand the single-step breed plan analysis offerings for other breeds across the UK and Europe, and also worldwide. It's a pretty key priority for us with the breed plan and here at breed plan. Okay, so single-step breed plan is really adding an extra source of information to the breed plan analysis for us to use. So if we think about conventional breed plan, which those breeds that don't have single step will be on conventional breed plan, or what those of you before you went to single step would have used to have. Conventional breed plan, we use pedigree information and performance information, both collected on the individual animal, but also related animals. And we use that to calculate the breed plan EBVs. In single step breed plan, we're still using pedigree and we're still using performance information. We are using genomic or DNA information as an extra source. So it's not replacing the need for performance recording. It's not replacing the need to have pedigree information on those animals. It is providing us with a third source that we can use to calculate those EVs. And what it does require is animals, or what we call a reference population. So that is animals with both phenotypes or performance data. So all of those traits that we've been talking about collecting in webinars 2 and 3. So your growth traits, fertility, carcass, other traits, all such as net feed intake, for example, all of those, they're the phenotypes or the performance data. And we also need in that reference population, those animals to have a genotype or a DNA sample. And it's really important when we are setting up single-step breed plan analyses that the reference population that we're using is representative of the whole population as much as possible. So we don't just want to have a small segment. It's genotyped and phenotyped. It's important that that reference population captures the scope of variation within breeds. So, for example, in many cases, the reference population to begin with will be made up of older bulls or well-used sires. It's important that they're from a range of different lines and from a range of different studs, so that we're capturing that variation. Now, that can be quite tricky depending on the breed. So it's particularly challenging for breeds with composite or diverse backgrounds. And so, for example, here in Australia, Brangus have gone to single step, but that was more challenging for us because they are a composite breed with a mix of Angus and Brahman. They're still, as far as I'm aware, open and allowing those parent breeds to be crossed together. So you have animals that range from majority Angus through to majority Brahman, and we needed to capture all of that variation. So that's challenging in a composite breed. Diverse backgrounds, I guess our example in Australia is Simmental. In Australia, we have the traditional Simmental population, like you can see here, but there's also black Simmental within the population. And so you need to manage the diversity across both of those. I'm not sure if that's the case for Simmental in the UK. I certainly had a quick look and couldn't find any black Simmental animals on the database. That doesn't mean they're not there. I just might not have had a good enough look. But that's the sort of challenge that animals with different types or breeds with different types can throw up. We need to make sure we're capturing those different types in the reference population. Okay, so at its basic level, genetics is really all about relatedness. So here, I've got my brother and myself with my parents. And if you think about what we inherit from each of our parents, both my brother and I got half our DNA from our mum and half our DNA from our dad. So we share 50% of our DNA with each parent. But the 50% of the DNA that we inherit is not identical. So my brother and I don't get exactly the same DNA from mum and exactly the same DNA from dad, unless the only exception there is identical twins. While you get DNA from your mum and your dad, on average with your siblings, you only share 50%, full siblings, you only share 50% of the DNA. And I think we understand that pretty intuitively, because if you think about your own siblings, or if you've got children, your own children, and you think about the differences between them, while there's certainly similarities, they're not identical. You can see that they might share traits in common with grandparents or further back, and it might be on a different side. So I think it's quite intuitive for us to understand that while we get DNA, from our parents, while there are similarities, we're not genetic clones. There is variation within the family, within the siblings. And exactly the same happens for cattle. So what I've pulled out here is a full ET sibling family. So we've got the sire up here and dam up here. And there were 10 full sibling ET calves, including these 3 bulls below. And you'll see that there are certainly some similarities between them, but there's differences as well. Some of them look a little bit more like dad, some throw a little bit more to mum. So there's variation within that sibling set. And so those differences in animals, depending on which DNA they've inherited, is what we're trying to capture with single-step. So single-step BREEDPLAN is really all about describing the relationships at the genetic level between individual animals. And so in conventional BREEDPLAN, what I've got here is the DNA coming through the pedigree. So you can see this individual here has got 25% of the genes from each grandparent. So it's inherited a blue and a red, and that blue has come from the paternal grandsire, and the red has come from the paternal granddam, and it's got 50% from each. And then if we look on the maternal side, the pink has come from the maternal grandsire and the yellow from the maternal granddam. It's inherited exactly half of each chromosome. And so it's 25% from each grandparent. That's what we assume in a conventional BREEDPLAN analysis, because on average, you share 25% of your DNA with each grandparent. But what single-step BREEDPLAN allows us to do is to say, well, we know that's not the case. When the animal is passing on genes to the next generation, the chromosomes don't come down in blocks of grandsire and granddam. They mix up a little bit. There's a biological process that mixes up the DNA from the grandparents in the sire, and that's what's passed on. So you don't get exactly 50% from it. You don't get exactly 25% from each of the grandparents. It's mixed up a little. And so what you can see here is exactly that case, where this animal has got a little bit more of the paternal granddam DNA than it has of the paternal grandsire DNA. And the same on this side, there's more of the maternal grandsire and less of the maternal granddam. And so what we're doing then in single-step BREEDPLAN is describing those relationships more accurately and understanding at the genetic level, what does it share with its grandparents. And so we can, what the single-step BREEDPLAN analysis can do, so long as the animals are genotyped, is it can work out the relationships between genotyped animals at the DNA level. And that's more powerful because instead of assuming that you've got exactly 25% from each grandparent, we're able to say, well, no, actually, in this case, this animal showing more with this grandparent, and that informs the EBVs. So it's really all about describing the relationships between animals at the DNA level. So what are the benefits of incorporating DNA information into the BREEDPLAN analysis? There's really 2 main ones. Firstly, EBVs can be generated for animals that do not have performance data. Now, there's a whole host of reasons why an animal might not have performance data. It might be an animal from a non-BREEDPLAN herd. It might be a young animal that's not yet old enough to be measured. As we talked about, I think it was in webinar 2, we collect those performance traits on animals from birth right up until they are about rising 2-year-old when we get our 600-day weights in our ultrasound scan. So a younger animal that's only 200 days isn't old enough to have had its 400 and 600-day weights, scrotals, and scan measurements recorded yet. Animals might not have performance data for traits which are hard or expensive to measure. So net feed intake is a prime example here. It's an important trait in terms of understanding efficiency of animals, but it is expensive. It does require animals to go into a feedlot and be fed for a number of days on feed, which has associated costs. Other traits which are hard or expensive to measure are things like methane, where animals are going once again into a confined space and having to there's equipment to measure methane, and they have to be repeat measured over a period of time. And so they're hard, they're expensive. We're never, well, I shouldn't say never, but we're unlikely to be in the situation where a large number of animals in the population are recorded. And that's where DNA is useful, because if some of the animals are genotyped and phenotyped for those hard to measure traits, we can generate EBVs for other animals in the population if they also have a genotype. Another example of traits that are only measurable in one sex. Obvious ones here are some of our female fertility traits and also mature cow weight. Once again, a measure of efficiency in the mature herd, but something we can only collect on 50% of the population. And also traits that are only measurable once the animal is dead. And the obvious example there is the abattoir carcass data. And then secondly, in addition to generating EBVs for animals that don't have performance data, more accurate EBVs can be generated for animals with limited performance information. So this might be a young animal that has its own performance data but no progeny, so a sale bull. And once again, most of those sale bulls are going to be rising 2-year-old, so they're old enough to have been had all of their performance data collected from birth to 2-year-old, but they won't have progeny yet, and so they generally will have lower accuracy EBVs than, say, an AI sire that's had a lot of progeny out there. So we can get more accurate EBVs for these animals if we incorporate DNA information into the analysis. But it's also particularly useful for animals in small or single animal contemporary groups. And I think it was last week, it might have been, no, last week was COP, sorry. It was webinar 3, where Boyd spent quite a lot of time taking us through contemporary groups and talking about how they can be maximised and how we compare animals head-to-head in BREEDPLAN. But we know that there are animals that will end up in small groups or in single animal groups, and they're often things like ET calves, because not many producers have large ET programs. Twins are an obvious example. We don't compare twins with singles, and most people, if they do have twins, only have a small percentage of twins out of the entire calving crop. And also breeders with smaller herds. So if you've got 20 or 30 animals, for example, while you can make BREEDPLAN work for you, you're typically going to have smaller contemporary groups than someone that might have a couple of hundred head. It's generally easier to get larger groups with more numbers. So those of situations where we can see more accurate EBVs being generated through the use of single-step BREEDPLAN. So why do we do all of that? Well, what we're really trying to do is give you as breeders a tool to make more informed selection decisions at younger ages. And that's really important, because if you can make your selection decision more accurately at a younger age. That means that you can shorten the generation interval, and that allows the potential then to increase the rate of genetic improvement, which is what it's all about. We want to breed the animals with the best genetics for the production system that they're in, and we want to do that as quickly as possible so that we can have the animals being as profitable for you as breeders and also for your commercial clients. Okay, that's the theory. Have we got any questions at this stage?
There's nothing has come in yet, Catriona, that I can see, but I'll keep an eye in case someone does put a question up for us.
Cool, thank you. I might keep going then. So that was the theory. Now I'd like to take you through a couple of case studies and look at whether or not single-step BREEDPLAN is delivering benefits for beef producers. So our first case study is Australian Brahman, and we're going to look here at EBVs being generated for animals that don't have performance data. So Australian Brahman were the first breed to introduce single-step BREEDPLAN. They did that back in April 2017. And just for a bit of background for our overseas audience who may not be as familiar with Australian cattle breeding as Boyd and I are, historically our Bos Indicus or our tropically adapted breeds haven't utilised genetic evaluation to the extent of their Bos Taurus or their British and Euro breed counterparts. And so in Australia, this is just showing you the location of our BREEDPLAN member herds across the country. Typically up here in the north, so Queensland, Northern Territory, in the top half of Western Australia, they typically have Bos Indicus or Bos Indicus composite breeds, whereas down south in the bottom half of WA, across into South Australia, New South Wales, certainly the bottom part of New South Wales, Victoria and Tassie, they are more typically Bos Taurus breeds, so those British and European breed types. Just to give you some context of where we're talking. So we're really talking about breeds up here for the Indicus. And so they haven't traditionally utilised BREEDPLAN and genetic evaluation to the extent that we see down in southern breeds. But each year in October, Australian Brahman have a large multi-vendor sale called Brahman Week. And so if we go back to 2016, the year before they introduced single step, and you look at the nearly 900 lots, we'll see that less than 20% of those lots had EBVs for the traits. So you'll see just under 20% had birth weight, 200, 400 and 600 day weights. That drops for scrotal, less than 15, or around 15% of the offering. And then for days to calving and our scanning traits or our carcass traits, we're seeing less than 10% of the sale lots of those 900 lots having EBVs available. Now, that's not a great thing, because if you don't have an EBV available on an animal, as we talked about in previous webinars, the way an animal looks and performs is a combination of its environment and its genetics. And if we don't understand the genetics and can't separate the genetics from the environment, then you're really not sure when you're purchasing the animal what you're getting in terms of a genetic package. But what we saw last year in 2024 was that over 90% of the sale lots, and similar numbers, high 800s, nearly 900 lots, over 90% of them had an EBV. And now, how has that been possible? It's really, sorry, I'm getting ahead of myself. And if we have a look at how that's happened, we haven't gone from 2016 having less than 20% of animals up to everything the next year. It has taken some time. So you can see there that 2017 they introduced single step and it started to lift, but it's really started to take off in the last 2 or 3 years. And the same if we look at 600 day weight, we'll see it's gone up over time, really lifted in 2022 and has increased since then. And it's really been driven by the widespread adoption of genotyping within the breed, including those that don't performance record. And I didn't explain this earlier. If we think about the Australian beef production system, down south where they have the British and the Euro breeds, they're typically more intensive production systems, whereas those production systems in Queensland and across the northern half of the country where they are running the Bos Indicus, they're quite extensive production systems. There are people that own thousands and thousands of acres. And so some of these animals will be running in 10,000 acre paddocks. So it's not necessarily that they don't want a performance record. There is some geographical consideration too in terms of the extensiveness of the production system that has made it more difficult than it would be for someone on a smaller property. So largely it's been this increase in the EBVs that we see has been driven by genotyping within the breed. A lot of people now in the breed will be genotyping animals ahead of the sale so that they can get EBVs in the catalogue, which has had a fantastic outcome in that the people buying those animals are now able to get a feel for the genetics and the genetic merit of those animals at sale time. And one thing I do want to say, because we're going to touch on this again at the end, is that this has really only been possible. The fact that they can genotype and get such an improvement in the number of animals with EBVs available has been possible because Australian Brahman has a large scale reference population. There are a lot of animals with phenotypes and genotypes. And that's really because there's research projects that have collected phenotypes and genotypes on a range of traits. They are long running industry projects. Genomics has been around for many years, and that project is collecting female fertility information. And then they also have a Northern Steer Bin project where the brothers of those heifers in the Genomics project are grown out and they collect carcass traits on them in the abattoir. So there is a robust population that has a lot of data, and they're all got genotypes as well as phenotypes. And I guess that really illustrates the importance of phenotypes. So I'm going to talk about that more later. Our second case study is for benefit 2, more accurate EBVs generated for animals with limited performance information. And so we're going to have a look at Hereford Single-Step BREEDPLAN for this. So Hereford Single-Step BREEDPLAN was also introduced in 2017. It was the first single-step analysis that we did that combined data from breed societies in multiple countries. And so those countries involved in this analysis are Australia, New Zealand, and Namibia. At the time of the study, they had been doing single-step for a couple of years. And so at the time, they had a reference population of 10,000. That was back in 2019. So it's certainly grown significantly since then. But what we did for this paper, which you can see down the bottom of the screen here, is we did 2 test runs. So we did one where the DNA information was included. So that's our single-step BREEDPLAN. And then we did another test run where we took the DNA component away and just ran the conventional BREEDPLAN analysis. And what's important to note is it's not that we did one in April and one in May, for example. They are exactly the same analysis with the same animals, with the same phenotypes. It's just whether or not we had the DNA component that changed. And what we saw when we did that is that the average EBVs have not changed. So there was very little change in the average EBVs for each trait. So you can see the conventional BREEDPLAN averages there for each one, and the single-step BREEDPLAN average EBVs there. So very little change at the population level if we look at an average. But we did see EBV movement for individuals. And so what we have here is their BREEDPLAN EBV in that conventional BREEDPLAN analysis, where we had no genomics. And then on the y-axis, we have the BREEDPLAN EBV in single-step BREEDPLAN. And you'll see that some animals have their EBVs have gone up, and some animals, their EBVs have gone down. And that's what we expected to see. If it was skewed in one direction, we'd be concerned, but we're seeing movement in both directions. And you'll see that there's some substantial EBV movements there in those young animals. So, for example, this one's increased quite a bit from where it was previously without single step, and this one has decreased from where it was without single step. So quite a bit of movement in the EBVs at an individual animal level, particularly for those young animals. If we look at the sires, which had a greater than 75% EBV accuracy in just the conventional breed plan analysis, you'll see that there's still movement. There's still some that have their EBVs have improved and some their EBVs have decreased, but that movement's not quite as pronounced as it was for the young animals. We also saw that the variation in the EBV has increased. So we've got more spread with single step breed plan. And that's important because it allows us to better identify animals at the extremes. And that top end are obviously useful for breeding programs. They're the ones with the genetics, the good genetics for the individual trait. And the bottom end, the ones that have spread more away from and their EBVs have decreased, they're typically the ones we want to cull for the breeding program. So it's allowing us to get a better feel for the extremes at either end. And the other thing that was really interesting was that EBV accuracy has changed. And so what we have here, again, is our EBV accuracy in our conventional breed plan where we don't have genomics, and then EBV accuracy under single step breed plan. And you'll notice that it's not a straightforward typical linear relationship. What we see is that those animals that have low EBV accuracy in conventional breed plan, when we add the single step information, the DNA information, you'll see they get a lift in accuracy. And you'll see animals that have gone from 5% accuracy up to, in this case, just under 60% accuracy with the addition of DNA. That's probably quite an extreme example. You can see animals here that have gone from 25% accuracy up to 40 to 50% accuracy. So we've got a lift, excuse me, in EBV accuracy from adding in the DNA. But you'll notice that if we look up here at the higher accuracy animals, that there's not such a pronounced lift in EBV accuracy. We might have an improvement of 2 to 3% accuracy. And that's not unexpected if you think about DNA information as being an additional source of information for the analysis to use. Excuse me. These low accuracy animals, or that have the low starting accuracy, they will have some pedigree information. They may have some own performance, but they've certainly not got progeny in the analysis. So for animals that are young and don't have a lot of information behind them, adding DNA is like adding additional progeny. It gives a boost in EBV accuracy. But if you consider an animal that's already highly accurate and it's got high accuracy EBVs, it's probably got pedigree, its own performance, and progeny in the analysis with their own performance. So we have a lot of information on those animals already. And so in that case, with genomics being just like adding some extra progeny, adding extra progeny when you've already got quite a large number in the analysis doesn't give you a huge boost in EBV accuracy. And that's exactly why we're seeing this pattern here. It's all to do with how much information did they have previously, how much more information does genomics give them. It's always going to give those lower accuracy animals more information, and that'll boost the EBV accuracy more for those lower animals. And we see this pattern repeated across traits. So that was 200-day weight. If we look at days to calving, you see the same pattern. These lower accuracy animals certainly got a boost in EBV accuracy by including the DNA information in single step. And these highly accurate animals, we didn't see such a boost. The pattern's the same. So I guess that's just two traits, but we did have a look at all of them. I guess the first lesson there is that the change in EBV accuracy is not going to be uniform across animals. Less accurate animals typically show bigger increases in EBV accuracy, when your condition included, than animals that have already had high starting ABV accuracy. And that pattern is consistent across traits. But how much of a boost they get when you include single-step on and include the genomic information really varies by trait. It's not uniform across traits, the pattern's uniform, but the actual physical increase in ABV accuracy is not the same. That greater lift is usually influenced not just by the heritability of the trait, but also by the number of animals in the reference population. So that's why we keep coming back to reference population. For single-step to be really effective, we need a robust reference population where we have large numbers of animals that are phenotyped or have performance information for that trait, as well as DNA. And this is a study, again, this is a few years old now, so I expect the numbers would have changed because these breeds have certainly increased their reference population significantly since this study was done. But one of their scientists at the Animal Genetics and Breeding Unit that does the breed plan development had a look at the average increase in EBV accuracy for animals that were 30% prior to the EBVs, prior to single-step being turned on, and looked at what the average increase was. And you can see across these three breeds, 200-day weight, the average increase was 20, 23, and 15. So in breed 1, if you had an EBV accuracy of 30% for 200-day weight prior to single-step, you on average went up to 50% as your EBV accuracy. And in breed 3, they went from 30% to 45% on average. If you look at days to calving, you can see that it wasn't such a big jump. It was from 30 to 33% on average in breed 1, and from 30 to 40% average in breed 2. And this is where heritability is important. Days to calving is typically less heritable as a fertility trait than some of our growth traits. And so we need a much larger reference population there to get the same level of increase in EBV accuracy that we would see for something that's more heritable. And the same for IMF or marbling, you can see that varied by trait as well. So it's really reflecting that reference population and highlighting why we do need a good, robust, representative reference population. So I guess what we can see there is it's working. We're seeing animals able to get EBVs that didn't have them previously, and we're seeing increases in EBV accuracy. So we're seeing more accurate EBVs on younger animals. But what it's really about is, do we see an increased rate of genetic progress? And I've got 3 breeds just to highlight what we've shown. So these breeds introduced single-step back in 2017. And this is their index value plotted by calving year. So average index by year, by calving year. And if we look at 2017, that's when they introduced single-step. So if we look at their index value from 2011 through to 2017 in this particular breed, $24.63. That's an average of $3.51 per year. But if we go from 2017 to 2023, index value increased by was $39.30, which is an average of $5.61 per year. And so, yes, the rate of genetic improvement has started to pick up. And if you look at this trend, you can see it's particularly picking up in the most recent years. We see a similar trend in breed B. So in the years prior, the average increase was $1.54 per year. It's now in this period here from 2017 to 2023, it's now $7.79. And the same thing, you can see that really rapid pickup in genetic improvement in the most recent years. And the same for breed 3. They were averaging $1.04 per year prior to introducing single-step. It's now gone up to $2.44, and there's a rapid increase in the most recent years. And so I think what we're seeing there is embracing genotyping since the introduction of single-step breed plan in their breeds. And if you have a look here, you can see that the numbers of genotypes are really increasing year on year. This is calendar year. And so you can see in all three breeds, we're seeing the numbers of animals with genotypes increasing rapidly. And anecdotally, we've seen a move from only genotyping sires, which is traditionally how we've set up reference populations because they're animals that are well used within the breed, to producers also genotyping their sale bulls and also genotyping their female breeding herds. So we're certainly seeing that the number of young animals that have genotypes increasing, and it's not restricted just to young bulls. There are a lot of heifers that are being genotyped now. Okay, have we had any questions come in, Boyd?
None at this stage, Catriona.
Okay, I will do our last little section just to finish up, but if you do have questions, please feel free to put them in the chat box. Okay, so I just wanted to touch before we finished up on considerations for beef breeders in the genomics era. Two main considerations. Firstly, should you keep collecting performance data? Excuse me. And then secondly, which animals should you consider for genotyping? So I think one of the biggest misconceptions out there as we've moved into genomics, the genomics era, is that you can stop performance recording once genomics is made available for your breed. Professor Mike Coffey at the Roslin Institute said this very famous quote quite a few years ago: in the era of genomics, phenotype is king. We still need phenotypes, we still need performance records. And that is because, as I've alluded to throughout this presentation, a reference population is really vital for the success of single-step BREEDPLAN. Single-step works best when your current population in the breed is closely related to the reference population. So we need to make sure that we're continually updating those reference populations over time, which means we need animals with both phenotypes or performance data or a range of traits, plus the genotypes or the DNA going forward. So it's giving us an extra source of information. It's not replacing the need to collect those phenotypes. And at an individual herd and animal level, genomics will likely give EBV accuracy a boost, but it's not moving young animals into the high EBV accuracy territory like you would see for a proven sire and AI bull. So really, you can consider DNA testing to be an additional source of information rather than the only source of information you collect. And the advantage of you collecting genotypes and performance data on animals in your herd is that means that those animals in your herd will become part of the reference population going forward. And that makes you closely related to the reference population. And that's important for getting the most benefit out of single-step BREEDPLAN. Which animals should you genotype? It really depends on what you want the genotyping results for. Are you doing this for single-step BREEDPLAN? Are you doing this for parent verification? Are you doing this to manage genetic conditions and qualitative traits? Or is it a combination of the above? So exactly what you're trying to achieve from genotyping will influence which animals. If it's genetic conditions, you might be interested in testing animals that have the potential to be a carrier so that you know whether or not you're doing carrier and carrier matings. For PV, you might particularly be doing animals that you're going to sell on to other herds so that you can be certain about their parentage. For single step, it's going to vary depending on which animals in your herd are already genotyped. You might have your sires done, and you might be interested in starting to do your young calves each calving year. But what we've seen here is certainly an uptick in the number of animals being genotyped since single-step BREEDPLAN was made available, and I showed you that earlier, but just to reiterate there again, we can see that each year more and more animals are coming in with genotypes. And anecdotally, we're seeing a shift from producers just doing their sires or their sale bulls to genotyping their female breeder herd. Now, we know that the cost of genotyping the entire female herd can be prohibitive. So one way to do that is to build up your females genotyped over time. and you could do your heifers each year. Now, when we think about the heifers, you've got animals that you're going to be keeping. So if you're going to be keeping them, they're a good candidate to do because they will hopefully go on to have many calves in your herd. They're going to be, that's going to be your female lines going forward. And so they're valuable because they're going to have future progeny for you. The maybes, they may be worth genotyping if you would like some further information in their EBVs and get more accurate EBVs on them through genotyping, as well as their performance data that they have in order to make a selection decision. So they're a potential to do. The culls, not so useful in terms of genotyping. You're not going to be keeping them going forward. So if cost is prohibitive, I'd be looking at doing the keepers, potentially doing the maybes, not worrying about the culls in terms of genotyping. And what this is showing here is just that if you are genotyping your heifers each year, you'll eventually get to the point where your whole female herd is done. So if we start in year 1 and just do the heifers that year, then in year 2, you do the new heifers, but you also have the heifers that you did the year before here and fully genotyped. And you can see that each year we add the ones in red, the ones in blue are already done. And over time, we get to the point where all of the older cows are already genotyped and you're just doing your heifer drop going forward. So that's an option if you're keen to start building up your females that are genotyped, but you don't have the capacity to do the whole herd in one hit. Start with the heifers and work through each year and build up numbers that way. If you'd like more information on that, we do have a technical note available. It's called The Case for Genotyping Females, and it's available from the BREEDPLAN website. If you go into the Extension Services area, resources and technical notes, if you can't find it, please let us know and I will find the link and send it to you. So I guess in summary, the ability to take a DNA sample and send to a commercial genotyping lab allows you the opportunity to use DNA technologies for a range of different applications, PV or parent verification, managing your genetic conditions, managing qualitative traits, and use in genetic evaluation. As we've seen through those case studies and looking at the trends, Single-Step BREEDPLAN is delivering benefits for beef producers. But really importantly, if you only remember one thing, please remember from today, please remember this: performance recording phenotypes are still vital in the genomics era. And that was all I had.
Well, thank you, Catriona, for that. We've just had a question come in. Yep. Does it matter if you DNA more heifers than bulls? Does that affect the figures?
No, I certainly wouldn't advocate just doing your heifers. I think there's certainly merit in doing your bulls as well, particularly the animals that you're looking to sell on. So your bull sale animals, for example. But I don't think that if you have more heifers done than bulls, for example, that it's going to impact the figures in terms of the way that selective recording would. It will certainly impact the figures for the individual animals because we have more of a feel for how they're related to other animals in the analysis, and that will impact the EBV. But I don't expect it to have a selection, a skew like we would see in selective recording.
Yeah, I agree entirely with that. What animal you do won't actually affect the figures, everything else being equal. But when you break it down, if you're talking about mature animals, for instance, a bull will have higher accuracy EBVs and therefore gain less from it because he'll have more progeny than a cow would at the same age. So yeah, you'd get a bigger benefit in the cow, but in terms of whether it's male or female affecting the figures, no effect whatsoever. We've also had another question or two questions. is there any percentiles in the genomic evaluation, and is there an international benchmark tool?
So as the first one, and I guess I didn't explain that clearly, sorry. Your breed plan evaluation, how it's presented to you, doesn't change with single step. There are still percentiles. We don't give you a genomic breeding value and a conventional breeding value. You get an EBV like you do now. The only difference is that the EBV will include DNA information as a source. So it's just still going to be those breed plan EBVs that you see now. It's just with an additional source of information. And so therefore, yes, the percentile bands will still exist, as will the breed averages. It'll be the same as you see now, just with the DNA information incorporated. Into benchmark tool.
Well, the reality is it's the same as what Catriona just said. It's the same as what you currently have as what you get from genomics. So it's still going to be within analysis results. So if you're a part of the Portuguese and European Angus analysis, for instance, it's still going to be part of that. You're still not going to be directly comparable against Australian and New Zealand Angus, for instance, because it's not the same analysis. That may change in the future if there's a combined analysis done.
Yeah, and I guess that's a good point. We'll talk about this a little bit in our future forward-looking webinar in the next fortnight. But you know, that's something that we certainly here at ABRI are considering, is how we can look at increasing opportunities for both worldwide or combined data sets where we combine regions. All right, if there's no further questions, I might launch the poll. So we've just got 5 questions and we would really appreciate your feedback on those. They're your standard feedback questions, but I've also included one. We're just interested to know whether or not you would find a future webinar on parent verification and or managing genetic conditions useful, just to assist us with planning for future topics. And I might also end the recording there, so I'll stop that now.